Good morning, students. Namaste. This is Dr. Uma Kiduvitan, Department of Food Biotechnology, Faculty of Agricultural and Veterinary Sciences, Jyoti Vidyapith Women's University, Jaipur. As you know, I am taking the course Metabolism and Bioenergetics Lab this semester. So we have already done the experiments on principle and operation of a spectrophotometer and determination of the amount of reducing sugars by Nelson's Homogy method. So this the third experiment is the determination of the amount of plants in a starch in plants. So for this, basically you will be, excuse me. Yeah. So your experiment is determination of starch in plant tissue. So for this, basically, we will be checking on the principle of photosynthesis. So you are aware that plants are the autotrophs in our, the primary autotrophs in our ecosystem. Like we need plants in order to fix to produce oxygen that they will be releasing into the atmosphere during the process of photosynthesis, and also using sunlight. Plants fix carbon dioxide into carbohydrates such as glucose and starch, and this carbohydrates that is produced by the plants are consumed first by the herbivores, and then the herbivores are consumed by the carnivores, and that is how the food web and the food chains develop. So what is photosynthesis? Photosynthesis is essentially the process by which plants are taking up carbon dioxide and then making it into glucose and starch. So the fixation of carbon dioxide by plants is the process of photosynthesis. This requires sunlight and is divided into two parts, the light reactions and the dark reactions. So in light reaction, there are actually like two photosystems involved, the P680 and the P700. And they are known as the photosystem 2 and the photosystem 1 respectively. And these photosystems, they get excited. They have molecules in them which can accept the energy from sunlight and go from a lower energy level to a higher energy level. And at this higher energy level, they will release the energy to another molecule, and thereby these excited electrons will come down to their ground state. And that is how these photosystems work. Now, once these excited electrons are leaving their energy, that will be accepted by molecules and converted and stored as energy in the form of ATP and NADPH. And this is the primary building block for glucose and starch. Now, you have all the ATP and NADPH that is stored by the plants. And this part, this production of ATP and NADPH from sunlight is the light reaction. And now you have the dark reactions, which is essentially the Calvin cycle. So, and also there are certain plants in the arid regions which can go for the C4 pathway rather than the C3 pathway, which is the Calvin cycle, and thereby produce like C3 will essentially end up with pyruvate, and C4 will end up with oxaloacetate or malonate, which are all 4-carbon compounds. So these pathways will fix carbon and thereby lead to the synthesis of glucose and then further into the synthesis of starch. But why light reaction and dark reaction? Light reaction essentially requires sunlight and this produces ATP and NADPH. So we could always say that like, yeah, dark reaction doesn't require light, but it requires the energy in the form of ATP and NADPH that have been produced by the uh, light reactions. So essentially the dark reactions do not happen in the absence of light, because first the light reactions have to occur, and then these energy storage molecules like ATP and NADPH have to be produced, and it's only after that that your dark reactions can happen. And the half life for storage of these NADPH molecules, which are the essential part of the dark reactions, are much lesser. So dark reactions also happen during the daytime only, when there is ample light, and light reactions are happening in the leaves where chlorophyll is present. So you know that in chlorophyll there are the thylakoid membranes, and the light reactions are happening in the thylakoid membranes, and the dark reactions will happen in the thylakoid stroma, the matrix of the thylakoid. So now what happens when you are keeping a plant in light and you are keeping a plant in dark? So during the dark phase, actually it is not producing any, there are no light reactions happening, so there is no energy source available, and so there is no glucose production, and there is no polymerization of this glucose into starch, which is the storage molecule for the plants as well. So this is not happening if there is no exposure to sunlight, but then it will be used up by the plants for its own energy purposes. So if you are like leaving the plants without light for some time, say like for 24 hours or something, the amount of starch present in those plants will be like nil only. And so whatever starch was there earlier also, it would be used up. And if we are giving water, then there is a chance that the plant will survive, but there won't be much starch present in that. However, if we are giving enough light, oxygen and water, the optimum conditions for a plant, then it will produce a good quantity of starch and store it. So the principle of this experiment is to figure out whether the starch is produced, like how much starch is produced by the plants in the presence and absence of sunlight. So if we are coming to the procedure, it's very simple. You take two plants. Yeah. So you will be taking two plants, and you will be taking beakers or glass jars, and then you will be using the saucepan and ethyl alcohol. These are actually these will be for the final stages where you will be using the iodine solution in order to check for the presence of starch. So take the two plants, and with that you will be placing one plant in a dark room for 24 hours, like totally without light, no windows, and the other one in a very sunny place, like a windowsill or somewhere where it will get ample sunlight. Like if you are keeping it in a summer season, then keep it preferably facing towards the north side, so that from east to west, the entire time that the sun is moving, there will be light available to that windowsill, and so you are giving that plant enough sunlight. And then wait for this 24 hours for this incubation period. So the plant kept on the windowsill will get light from morning till evening, and after that it will be dark period for that, where the plant will get rust, and the one in the dark room will not have any light, and so no light reactions and no synthesis of starch is happening in that plant. And now what we will do is, we will keep the beaker in a saucepan full of water, and then it will be filled with ethyl alcohol, and then we will heat up the ethyl alcohol until it begins to boil. And then out leaves from each of these plants, and you will be dipping it in hot water for 60 seconds using tweezers, and then dropping the leaves in the beaker for a couple of minutes, like until all the chlorophyll has been leached out from the plant, and then from the leaves. And after that, you will mark petri plates, and you will be keeping these leaves in those petri plates for some time. And then after that, you will be adding droplets of iodine onto those leaves. So what is happening over there? So that is the basic principle of this experiment. Like the hot water actually will kill the viable contents of that leaf, and once that is happening, it will break down the chlorophyll and remove all the green color, all the unwanted pigments out of the leaf. And then what is left will be the starch, because starch is a polymer, so that won't be leached out of the plant so easily. So the major components that will be left in the plants will be the structural components and starch. Now, if you have the two leaves, what will happen? One is from a leaf that has been kept in a leaf from a plant that has been kept in dark. So no starch has been produced in that leaf for the previous 24 hours. So, and you know that iodine is a brownish colored liquid. So you are adding iodine onto this white leaf. So another purpose of this is when the chlorophyll is leached, you will be able to observe the color of the iodine on the leaf very properly. So if you are adding the brownish colored iodine onto the leaves that doesn't contain starch, it will be brownish in color, because the iodine is there on the surface, but it has not converted into brownish, into the blue black color of after reaction with starch. However, when you are doing this with the leaf of the plant that has been kept on light, like which has been exposed to sunlight in the previous 24 hours, then what happens is there will be ample amount of starch in that leaf, and this starch will react with the iodine, and so the brownish colored iodine that you had put on the leaf will turn the leaf into a bluish black color. And so you will actually get, this is your control which has got no starch, and so it will be brownish in color due to the iodine solution, the unreacted iodine solution. And also there will be the leaves of the plant that was kept in sun and which has got starch in it, and so it will react with iodine and give the blue black color. So that is your experiment. So what is your procedure? The procedure is you will be first incubating your plants under the separate conditions. So one will be in dark room for 24 hours, one will be on a windowsill for 24 hours where it will get light for about 8 to 12 hours, or rather say 8 to 14 hours, and then it will get dark period for the rest of the time. So that will be like around 16 hours or 16 hours, or maybe if you are 10 hours to 16 hours, it will get dark period as well. So once that is done, you will be taking the leaf and first putting it in boiling water. This boiling water will kill the leaf. And once that is done, it will also make it easier for the ethanol to extract all the components from the leaf. And once that is done, you will be putting it in ethanol, separate in ethanol for 2 minutes, boiling ethanol for, or rather ethanol that has been like heated to boiling point. It will be put for 2 minutes. Then what happens? All the chlorophyll and other pigments from the leaf will be leached out into the ethanol, because ethanol is an organic solvent. And so these pigments are also quinones and all, which are which have high solubility in organic solvents, but have very less solubility in water. So these will all be leached out, and what you will be getting is a white colored leaf. This white color background will also make sure that the results of your experiment will be very clearly visible. And once this is done, you can mark your Petri plates into which you will be placing these leaves, as the one for the plant that was kept in dark and or the plant that was kept in light. And you now have two leaves, two white colored leaves. You will be adding a few drops of iodine, which is a brown colored liquid, onto both of these leaves. And then you will wait for some time to see the color changes happening. So your result will be that for the plant that was kept in dark, since it doesn't have any significant amount of starch in it, it only has the structural components of the plant. It doesn't have any starch. And iodine doesn't react with cellulose, which is the structural component of plant. It only reacts with starch in this way to give a blue-black compound. So once iodine is added to the leaves, the leaf of the plant that was incubated in dark for 24 hours, iodine will not have any color change. So it will remain the brownish colored product, the brownish colored liquid. It will, or rather it will retain the brownish color of iodine as such. Whereas for the plant that was incubated on a windowsill for 24 hours, there will be ample amount of starch. And so depending on the amount of starch that was produced, you will have a bluish black color developing on the leaf, and that will be visible throughout. So with that, you will be able to identify between the leaves and also figure out the production of starch in leaves that happen on exposure to sunlight. So that is the basic principle and the procedure for this experiment. So your result will be an image of the two leaves, or you could draw the two leaves and show the color change in them, and then mark which is which. That will be your expected result and the outcome of this. So your course outcomes for this experiment, what is expected is that you identify the principle of photosynthesis, what is happening in photosynthesis, why starch is being produced in one leaf, in one plant, whereas starch is not being produced in the other. And then you have been given the procedure and your expected results. So that will be it for this session. Hope you have understood it. Thank you. Have a good day.